This is Our Voices, Our Legacies, the Medical College of Wisconsin's Oral History Project. Today is March 8th, 2017. I'm Dick Hatchke, MCW's Chief Historian. We're talking today with Dr. Rodney Willoughby. Dr. Willoughby is a professor in the Department of Pediatrics in the section of Infectious Disease. Thank you, Dr. Willoughby. I appreciate you taking time.
It's my pleasure to be here.
Great. What led to your decision to become a physician? When did you decide you wanted to go into medicine?
Well, there's actually a long family pedigree of physicians in my family. Nobody in the immediate family, but my grandfather and my uncle were both physicians. And we'd actually had physicians going back apparently to the American Revolution, so it was genetically determined.
And you went to Johns Hopkins for medical school.
Yep.
That's one of the nation's most prestigious medical schools, so it was an honor to be accepted there.
It was. It was the only place I was ever accepted to. So
At what point did you decide that you wanted to go into infectious disease?
Well, I'd grown up in South America, and so the idea of working for the developing countries where really most of the world's under 18, it was clear to me that I wanted to be a pediatrician, and most of the communicable diseases are still communicable in that part of the world, so this was, to me, a no-brainer based upon my background and how I'd been raised.
Mm-hmm. And why pediatric infectious disease?
Well, again, actually during medical school I took off a year and I worked in West Africa, and there it was clear that really half the population's under 18. They were the future of the country, and that they were probably the most important piece in order to have those countries develop well. So that's what I decided. I decided to be a pediatrician at that time.
And where did you did your residency training at Johns Hopkins as well, correct?
I did it actually partly at University of California, San Diego, and partly at Johns Hopkins.
Mm-hmm. And after you finished your training, what were your first jobs?
Well, from resident, I became a fellow. I was actually hoping to do a research fellowship. That actually fell through, and I ended up being a clinical fellow. And then I got a grant to let me continue for a couple more years. And at that point, my lab work suggested I needed to learn one of the courses I hated the worst, which was biochemistry. And so I ended up doing 5 years of postdoctoral in biochemistry. So my real job was really probably well into my 30s. And that was, again, being a clinical infectious disease doctor, but also running a laboratory.
And what was the focus of your research?
At that time, I was actually very much interested in rotavirus, which is a major cause of diarrhea. It kills 800,000 kids annually. The vaccines had not come into play yet, and so this was a major problem. And so I really was not the life of the cocktail party circuit. I worked on diarrhea.
What was the focus of your research?
I worked on rotaviruses, and at the time the vaccines were not particularly successful. So I was actually looking at alternative strategies were based upon some work actually with what are now with enteroviruses, in that they would have, say, 90 different viruses, but all would have the same receptor. So there was a lot of power in going after treatments which could mimic receptors. Very similar to now what's been done decades later for HIV. But I was very much interested in that at the time, and so I spent my time learning carbohydrate biochemistry and cell surface biology. So.
Mm-hmm. What led to you coming to Milwaukee?
Well, I was looking for a lateral move. I'd run our division really too young for a number of years, and also at that point had several young children, and it was a very expensive place to educate my children in Baltimore. You really had to go to private school, and so I was looking for a change in venue and looking for a place where the public school system was really good. So this was also one of the few places that wasn't a fixer-upper. Usually people move laterally when there's a problem someplace and they have to rebuild. This was running smoothly. There was a succession that was being planned, and so I came here with that in mind.
Now, this would have been around 2004, I believe?
2004, right.
And who were some of the people that you met with when you first looked at MCW as an opportunity?
Well, Joe Chusad is still my division director. He's still threatening to retire. And Peter Havens, and I actually knew of Peter from my HIV work. He was nationally known for really reducing the transmission of HIV from mother to infant in the state of Wisconsin, which is still basically a leading star. And then that time Bob Kliegman was the chair of pediatrics, and he was one of the principal editors of one of the main textbooks of pediatrics, Nelson's. And at the time, Colin Rudolph was here, and he was head of GI, but he, of course, was one of the main editors for his father's textbook of pediatrics. And so you had a lot of really smart pediatricians writing major textbooks out of here, and that impressed me greatly. And then finally, at the time, Nita Saltzman was here, and she was looking at the microbiome before it became popular. And I'd been doing research on cerebral palsy as a possible infectious disease, and my work was moving towards the microbiome of the vagina and the uterus. And when I came here and found out she was here, she was really a similarly interested soul. So I came here thinking I would work closely with her, which lasted all of about 6 months, and then I got distracted. So.
Now, Starkey Davis had been the chair of pediatrics before Dr. Kliegman.
Yes.
He was in infectious disease as well. Was he somebody that was still around at that point?
No, Starkey had gone. I actually met him subsequently as a great man size-wise too. He was a very famous football player of his time. But at the time, I really only met him during sort of an annual professors conference where we bring in a guest speaker, and he and his wife would come back and attend the lecture, and we'd go out to dinner and stuff. So that's how I met him.
Now, when you first came on board, what were the kinds of patients that you were seeing before Gina Giese changed everything for you? What was the norm in terms of the infectious disease? What types of problems were you encountering?
Well, the infectious disease world is pretty much still the same now as it was then. We see a lot of critical care patients with complications, infectious complications, or sometimes severe infectious disorders, which land you in critical care. There's always been a lot of immunocompromise and unusual bugs, and so the hematology, oncology, bone marrow transplant group here, and the immunodeficiency group here has always been strong too, so we get to see some of those. And then this hospital is really interesting because it's very good clinically, but it's really in some aspects is a glorified community hospital. There's a lot of just bread and butter general stuff, and so we see a lot of bone and joint infections. We see a fair amount of meningitis or encephalitis, which are particular interests of mine, skin and soft tissues, pretty much a little bit of everything. So it's a very nice, balanced general practice with a lot of new stuff, not just a lot of referrals of very exotic things, and that's great fun.
Now, you mentioned that your career here changed about 6 months in. Tell us what happened.
Well, it's actually the second time I was on call here, so I really knew nobody, and I got a call from a transport physician who ended up being one of the other people taking care of it. It was Nancy Ganayam. She's no longer here now. And they had a patient being sent down from Fond du Lac with encephalitis, and they were going up to pick her up at the time, but their primary physician, Dr. Dono, had come by on Monday because he'd heard that she was getting worse in the hospital. And not knowing what she had and knowing she was getting worse, he did what all good physicians do, which is to retake the history. And when he did so, he learned that she'd been exposed to a bat bite about 3 months earlier. And so he called Dr. Ganayam back saying, I just heard that there's been a bat bite. Should we be thinking of rabies? And so then I got the call saying, what should we do about that? Should we put our patients, put her in isolation for the transport? And I answered in the affirmative. And then I told them that we could pretty much have the disease excluded within a day or so by just being prepared when she got here. So we had it all set up so that when she would arrive, we would do all the biopsies and tests necessary. And we should have an answer back in the next day or so, and then we could move on with whatever else she was ultimately going to have, which almost certainly wasn't rabies.
Mm-hmm. Now, this was 3 months post-bite.
Yes.
And so given that length of time, that really limited your options then, didn't it, in terms of any kind of treatment that could be provided?
Well, there had been no survivors of rabies at all ever with any kind of treatment, so our options were very limited. The long incubation period for rabies is one of the characteristics of rabies, and is what Louis Pasteur had recognized, and that he could actually generate an immune response with a vaccine faster than the infection would proceed, so you could catch up from behind. And that still is the general approach to the world, that we wait for the exposure to happen and then we give vaccine after the fact and it can essentially lap the virus and prevent disease. Because rabies was uniformly fatal, I actually knew almost nothing about it other than the fact, because it was really futile to know more because you couldn't do anything about it. But I'm a great believer in the telephone as a modern piece of technology, and so when she was looking more and more like she might actually have rabies over the course of that first afternoon, I called the CDC up and asked them if there was anything new coming along. Typically there's a delay of 2 or 3 years between the science and the publication, and so they confirmed there was nothing new, and I then started thinking about, well, what could we do since the conventional stuff had regularly failed? And that's kind of where I think we diverged from what was normally done and where we got, in retrospect, very lucky.
And so what was the decision? What was the course of treatment that you tried?
Well, what had been noted for probably close to 20 years is that there had been a lot of near misses. There had been almost survivors that did very well for a period of a couple of weeks, and at time of demise that they no longer had the virus in them, that they died of a medical complication, but that there was really nothing, there was no virus anymore, and that everything that I knew how to do had failed miserably. And so the concept was that this disease must somehow be killing the patient just by the brain's malfunction, that this was a disease where if you could buy time, then you could clear the virus in a couple of weeks. And so, and it seemed so obvious that there had to be a flaw, and that's what actually gave me the greatest pause. And we figured out how to get around that, and then we decided to essentially just suppress her brain function long enough for her immune response to come in to do the job. And to my great surprise and relief, and with a lot of bitten fingernails in between, the strategy ended up working, and it's now worked another 14 or 15 times. So.
Who were some of the other people that you worked with at MCW and at Children's that were collaborators, people that you were able to talk things through to see, is this really the right approach?
Right. Well, the main issue was when you invent things in medicine, usually you cause harm. And so my greatest worry was that we would actually harm the patient. And so I asked Joe Chusid and Kelly Henriksen, who's another virologist in our group, to join me in a meeting. And since I didn't know anybody, I asked Joe if he would bring in a couple of more people. And the idea was to have enough experts where we could get a consensus, and if we had to blackball the idea, which was a simple one, really it's just using anesthesia like you would for any surgery. Like you put the patient down for a couple hours and you bring them back up again, and you avoid all the dysautonomia in between. So I asked for 2 neurologists, and one was a neurovirologist named Kate LaFond, and the other one is still here, and he was an epileptologist, and so was used to putting people under in order to control their seizures. And so we had 2 neurologists, and then we had Nancy Ganayam, who was an expert critical care doctor, and we had a second critical care doctor as well, who was also on service at the time, and then really just bringing in, so as a ringer, is George Hoffman, who's a close colleague of Nancy Ganayam's, and he was considered to be sort of a premier physiologist that really knows way too much and knows how to use it, and so he's brought in sort of as our ringer, and then and that was the group, and the idea was that we would convene if the diagnosis was established, and we would come up with a plan, and they had the heads up as to what the disease was, and so. When the results came back within 24 hours that she, in fact, did have rabies, that was 4 o'clock or so. By 5, we had everybody in the room, and by 6, we were out of the room, and we'd discussed sort of 3 options, one of which was to palliate, make her comfortable, maybe send her home, which is really what's done in most of the world. The second option was to do everything, antivirals, which had never worked up to that point. And then the third option which we decided we'd only offer if the first two options were not attractive to the family, was to consider just sedating the patient long enough for the immune response to come in. And Gina's parents, John and Ann, are smart people, and they didn't like the first two options. And then, to their credit, they decided that, well, why don't you try something new, and if nothing else, you'll be closer for the next patient. And so they essentially accepted that she would die. They had tremendous faith and were hopeful that she would not, and I think really had faith that she would not. But then essentially released her to us so that we could learn something for the next time. And boy, we learned a lot.
You know, it seems like it moved pretty fast in terms
Very
of
fast.
the group that you pulled together, the decisions, the process. Did that rapid process of getting a consensus and deciding this is the course of action and getting the consent from the family contribute to the successful outcome?
You know, I think if you get the right people, they're all pretty much prepared ahead of time. And so you can get from A to Z pretty fast, you can get going. And when you do that quickly, you mobilize and you move quickly. And I think that actually does contribute a lot to better outcome. The risks were enormous. We understood failure was probable, but we had a decent theory, and we decided to go, and then everybody basically pitched in, and we went. It's funny because it was both fast and slow. I've always been an athlete. The entire period when we treated Gina really felt like being in the zone, where when you're in the zone athletically, time slows down, and it feels like you have forever to do stuff. And even though it's happening at split-second speed. And the 2 weeks we took care of her really had that feeling. There was really never doubt, which is built into most consultants, including myself. But everybody was pretty much worried but certain. And it went both fast and slow at the same time.
Now, rabies is not that common a disease. Was this the first rabies case that you had encountered?
This is the first rabies case I'd ever seen. In fact, for the boards, you only need to know rabies is fatal. So, yes, it was the first. But you know, I think sometimes you put on the earth for a reason. And in retrospect, with a little more time, once the 70 days had gone by and she'd gone home, it became clear that there are a lot of aspects of my training which had kind of led up to this moment. You know, I'd trained as a biochemist. There's a lot of scientific method to this. I was working on cerebral palsy as an infectious disease, so I understood to some degree brain damage and inflammation and why therapies work and how cytokines work differently in the brain than elsewhere. And so I'd been trained as a clinical trials person. There's just a lot of skills which had not been put together, but which I distractedly done at different parts in my life, and they all kind of came together for Gina, really. And so it was a delight to be able to put it all together, and it's continued to be a delight to sort of learn. I really feel like I've been through medical school all over again since Gina came.
So after the decision was made to put her into this induced coma, how much time period? Was it 70 days you said then passed?
The plan was to put her down for 7 days, which is when the immune response should be in. And the plan was, given the fact that our thought was that the brain kills the patient, was to really leave her alone. So the first night was pretty sleepless. They got her down, and I was in the library pulling every clinical report I could find on rabies with the idea of mapping the complications by day. And so at, I don't know, 2 o'clock in the morning, whatever, I had a type list on the wall with what would happen by day and what our responses would be. The responses, of course, I was helped with by the critical care doctors. And then we proceeded to just drive it by wire. You know, we weren't using exam at all. We were just following her, and then every day we could sort of cross off complications that had come and gone without it. There was a lot of discussion. There are always more naysayers than there are positivists, and so you had to play defense. And so I got used to playing defense twice a day on morning and evening rounds, and I did that pretty much for 70 days. But there were always people in my camp. You know, George Hoffman and Nancy were there a lot. They helped me to fend off some of the other Kelly Teves too, some of the other critical care doctors. And we just kept going, and again in the zone, just like we could do no wrong. In retrospect, we did a variety of things right for the wrong reasons. So it's always better to be lucky than smart, I think.
You also had the support of the hospital in this process too, didn't you?
I think the support of the hospital actually came much later. They actually committed. Within a few months, we actually got a call from Marines that were on a beach in Haiti with a potential rabies patient. And at the time, I think we were pretty much doing conventional critical care on a conventionally ill patient, and the hospital wasn't involved. But after the fact, both MCW and Children's Hospital stepped up when it was clear we had our first, and it was obvious we were going to get phone calls. And at that point, the hospital really thought through as to what they'd be willing to, who they'd be willing to accept here, and what the limits of that would be. Again, large financial exposures. She cost, I think, about $800,000 to care for her. That's a lot of money. And again, MCW committed some space. I actually got some advance funding to get some equipment in pretty quick, within a couple of months, to be able to start the research side of it going. And MCW has really supported the website ever since, which has been used I don't know how many times to download our protocol and for other patients to be treated.
Are there other centers in the world that have had a volume of rabies patients where there would be experts in other institutions?
There are a variety of places in the world where rabies is seen more commonly than here. Interestingly, in some cases they're experts there, in other cases they're really not. The usual consensus on rabies is to let them die quickly and as painlessly as possible. And so that doesn't provide a lot of clinical expertise, maybe in the diagnosis, but pretty much the disease sorts out within a couple, 3 days, most of the world. By the time you show up, you usually die within 2 days of being hospitalized. Thailand has a couple of real true academics that specialize in disease, and they've continued to publish extensively on it. They're not always great supporters of ours, but they are careful thinkers, and they are as highly motivated as we are here. There's some other people that are more laboratory based, that are clinicians, and again are quote experts in rabies, but it's really more on the lab side of thing. The Philippines has a lot of rabies, but for whatever reason, there's just never been the interest in treating rabies there. Much more interest out of India, where probably half the world's rabies is concentrated. That's been late to the game, but that's now happened. And then China, depending upon the winds of the politics at that time, have continued to be interested and have continued to have research efforts, lab efforts involving rabies.
When you were working with Gina's case early in this process, were you able to collaborate or contact any of these other institutions to see if they had input, or again, was it just there's no cure?
Well, the funny thing was we got a lot of great support, and continue to do so, from the Centers for Disease Control in Atlanta. They are probably the preeminent rabies lab. They have superlative diagnostics, and we've continued to have really almost monthly conversations as cases crop up someplace in the world, or at least in the hemisphere. So they've been great help. The problem with the research field is the research field was so biased towards vaccine development that there really was no work on natural history and zero on rehabilitation. Even though there had been animals that had survived fairly predictably, rarely but predictably, but those animals are always sacrificed and you went on to the next experiment. And so when we were having Gina recover, she actually had a variety of clinical challenges to us. She had movement disorders, which were very troublesome and which we had no idea. And she was recovering in a way which to me looked like sort of the repeat ontogeny of neurological development in a human, you know, from baby on up. But there was no one to talk to. I ended up talking to a lot of neuroanatomists that use rabies virus to map the brain, and they were interested but really had not the clinical correlation in terms of movement disorders, for example, to be helpful. And no one has studied rehabilitation of rabies. And so we were so blind. We actually were blind for probably 5 years, where we had no idea how she was going to recover and what the next challenge was, or even the great fear as to whether she might relapse or somehow do something new.
So it was beyond just the Milwaukee protocol. Every step of the way you were in uncharted territory, and so in terms of rehab and everything else, it was a whole new situation.
It's a very uncomfortable position. You know, I have much more empathy for people that are the first one to do a particular surgery. Even then, you get to practice a lot before you do it. This was like literally having something land from a spaceship, and you had no idea what it was or how it would play out or what the late repercussions would be. And it's a very uncomfortable feeling because you're really trained to be a scientist and depend on data, but you never think you could be the one that's going to generate all the first data, and that's uncomfortable. The great comfort is I like seeing associations, and as challenges came up in rabies care, which have continued to be the case, you can almost always pirate treatments from similar medical conditions. And so as long as we could recognize the complication, there was almost always some other medical analog where we could then go and talk to them and ask them, well, how do you treat this or how do you treat that? For example, vasospasm, which is sort of spasm of the cerebral arteries in rabies, we predicted that based upon some biochemistry we had. We confirmed it with sonogram non-invasively in a variety of patients around the world. And we then looked at subarachnoid hemorrhage, which is a common bleeding of the brain that happens in older adults. And there's a whole not perfectly evidenced battery treatments, but where people do it regularly, and there are protocols, maybe not fully evidence-based, but they're there. And so you can glibly steal their protocols and apply them to rabies. And that's how I think we've continued to advance, is largely by being thieves. We just take various pieces off the shelf from other diseases and plug them into rabies and see what we got. So.
Gina made a remarkable recovery, and some of the neurological problems that were first identified were things that she was able to surmount. How is she today?
She's great. She's now a mother of twins. She's probably a little more sleepless than she'd like to be, and we'd like her to be. She obviously has toned down. She's been very active in the rabies field, largely education side of things. But she's doing great. Really, you can't tell that she has anything wrong with her anymore at all. There's probably something under there, but it's not evident. And she's now just a normal Wisconsin mom with a set of twins. So it's been delightful. I don't think it's a closure. She's on a mission. She feels like she survived for a reason, and she continues to again educate and advocate. And I expect to see more of that as the kids get a little bit older.
Is it accurate to say, though, that every milestone in her life, in some respects, is also adding to medical knowledge? The fact that she was able to give birth, the first rabies survivor to do so, I would imagine.
Well, yes and no. I actually think she's largely one and done. We've had some challenges where she's gotten bit by some other animals she shouldn't have and stuff like that. We've been reluctant to immunize her just because she's had huge titers. She shouldn't need it. But also we worried whether we might get some sort of cross reaction or some adverse effect from the vaccine, given the fact that she has such a major response to the real wild type virus. And so that way she's been a challenge. But this is a virus which, to our knowledge, doesn't live in you forever like many other viruses do. And so having survived, she's done. rabies doesn't course in the bloodstream, it's only in nerves. And so there are probably a dozen children out there who were born of mothers who died of rabies. And so the concept that you can have normal children during rabies is clearly established. So, and she's actually not my only, hers are not my only rabies grandchildren. I also have a rabies grandchild in Peru at this point from another survivor down there. So I think she actually continues to offer her blood, for example. She offered cord blood on her infants at the time of birth if it would be helpful for science. I don't think we have the scientific questions. We don't know how to use her offers yet. So I keep telling her thank you and we'll be in touch. But to this point, absent maybe total genomic sequencing, which is probably soon to be, we don't have much use for her other than in the public relations side of things.
After you went through the whole process, then you published. Where was this first reported in the medical literature?
Well, the first report in the medical literature actually was in the public health literature. The Centers for Disease Control have a rapid communication for communicable diseases called MMWR, and it's a weekly report. And so that came out there pretty quickly, and that really alerted a lot of people. There was also a whole chain of daisies among rabies labs and rabies researchers around the world, so that while Gina was under care, there was really daily well wishes and prayers from a variety of scientists in the rabies field. And so there was sort of this informal knowledge of what had happened. And then we had a little bit in the lay press from John, who would talk to the garage station attendant every night, and then he would turn and talk to the local news, and we weren't allowed to acknowledge that she existed, but the newspapers seemed to know everything that happened. But we finally published for real in New England Journal of Medicine, and that was a nice short report. It came with an editorial by probably my leading adversary, who remains so, and that got it a lot of attention, which is probably correct. Probably should have happened.
When you mentioned adversaries, there were those who questioned whether this was.
There still are.
Which is healthy.
No, I agree. So people actually ask me all the time because it's a small group in the rabies field, so the interpersonal exchanges aren't always collegial. But at the same time, again, science is about challenge and justification and thinking through the arguments, and that's been vigorous for the last 12 years or so, and it actually gives you something to lean against. It certainly has kept us very transparent and honest in how we count cases and how we report.
After Gina, then there were other cases where you were brought in to consult and where the Milwaukee protocol was used. What was the success rate?
So we're quoting about 20% success, so about 1 in 5 patients survives. This is really only with critical care. We learned that the drugs that we thought we were using as antivirals had no effect. We think that's about to change as of this year. But it's been 12 years of essentially doing conventional critical care. And I think what we do far better than other centers treating rabies anywhere else in the world. And I think the reason is that we've been careful observers. If we see something 3 times, it's real, and we prepare for it. And so at this point, I can pretty much tell you to the day what's going to happen to your patient for the first 2 weeks. And we can tell you what to do about it. And that seems to lead to a survival benefit where we've got 20% survivors and pretty much nobody else has any. But it's not great. 20% is nothing. A lot of cancers are in that range. I actually take inspiration from those cases. I talk to our oncologists here. You actually 20% allows you to survive long enough to be able to measure things in the lab and to know what the complications are. And When I was a resident, my professors were among the first to treat, say, acute lymphocytic leukemia of childhood. And at that time, it was uniformly fatal. And I've gone back and read the tremendous clash in the medical literature at the time about how it was unethical to treat children with cancer, that all you did is prolong suffering and that no one would survive, and this is well known, and that the people doing this were brutes. And the defenses were that they could show that year by year they got better and better. And now, probably 10 years ago, there was a lovely article where the survival rate was 85% all comers, which is a lot better than zero. And the drugs used had not changed in the ensuing 20, 30 years. They were just used better, and people knew about infections and other complications that came. And so you just got better over time, even though the same armaments were being used to fight the disease. And so that, to me, has been one of two sort of progress in medicine stories that I use repeatedly in defending what we're doing with rabies as we move from 0% to now 20%. And again, finally this year we have some new antivirals, and I think we'll probably get up into the 50s. I hope so.
Now, Gina was in her early teens when this happened. The animal that bit her was a bat. Does it matter what the animal is? If it had been a raccoon, if it had been a rat, is that something that may create a different situation? If she had been in a middle-aged or older, does age come into play? Are these things that have been considered?
Well, one of the nice things about getting a protocol and going over time is you can actually figure these things out, because no one actually, physicians are like cats, that you can't hurt them anywhere, and so everybody wants to change the protocol, make it better. So there's a lot of variation, and then in resource-poor settings, a lot of times you can't do everything. So theoretically, under age 5 and over age 50, your immune system is lousy. We require you to do all the heavy lifting. We have only critical care support for you. And so, and our data support the fact that under 5, we just got our first exception. We have a 2-year-old now in Peru who just survived. But before this, under 5 didn't work. Over 50, it didn't work. That's predictable. So in terms of age, I think that's absolutely the case. In terms of dog versus bat, for example, the two big groups, that actually wasn't known because no one survived long enough. The supposition was that bat was milder, and that's because it was more paralytic. That's probably actually just the opposite. Bat actually seems to be more inflammatory. The higher inflammation limits the disease to your spinal cord, and so you die still 100%, but you're paralyzed as one of the causes of death, whereas the dog one, being less inflammatory, tends to get further naturally. What we've seen, though, is that bat rabies tends to get a lot of brain edema, so swelling of the brain, as the immune system comes in, and generates really too vigorous an immune response. So ironically, we now actually try and suppress the immunity of patients with bat rabies. Dog rabies, on the other hand, tends to be, it replicates in muscle, which bat rabies does not. It tends to replicate, or at least cause disease in heart muscle. And so we get heart block as a late complication in about 40% of dog rabies. We never see it in bat rabies. So we see different immunological responses. We see different organs affected. They still overlap, but they're clearly recognizably different. We have only a smattering of other lyssaviruses, rabies cousins, if you will. We have a smattering of raccoon rabies or fox rabies and the like. One of the other problems is that rabies from bats can actually then get into a carnivore and transmit. So we have a little series on bat rabies in cats, for example. And that we have yet to get a survivor out of that group, maybe one, who died during rehabilitation, but who we consider a survivor. But those seem to be different diseases too. So there are a lot of flavors, and I think it remains to be seen how different these things are.
Rabies, again, is not a common disease. Did you find that getting funding support for research was difficult, or were people excited because you had made such a significant advance?
No, actually, funding's been very hard. Rabies is actually so neglected that it's actually not counted among the neglected tropical diseases. So even though there's a special effort for them, there's not a special effort for rabies. When really statistically, if you have one, you've already proven the point. We actually talked with the NIH about whether we could get a clinical trial going to see if we could replicate it. At the time they said, well, do 2 or 3 and then come back to us. We then came back with 2 or 3 and they said, well, you need more data than that. Maybe you should try and get an animal model. We wrote for animal models. The animal models are high risk by study sections. They tried to do something with internal funding, which they couldn't even get the animals to survive without rabies long enough to have it be a survival model. So I've had one 5-year grant out of the NIH as part of a joint experiment where that's finished recently. The data we're still analyzing. It's been slow to come by because rabies treatments are relatively rare, so we've taken longer than we'd hoped to get our data in order. But I'm actually working on that today, so it looks good. But it's been pretty much hand to mouth. We have one family in particular out of Texas who lost their only child to rabies. They have donated to us consistently, and we use their funds to partially defray my technician and to fly in rabies samples from around the world when and if we can get them. But other than that, it's been pretty much a labor of love, or really labor of hate. This is one ugly disease, and I can't wait to get rid of it.
What's the focus of your research right now on rabies? What is it that you're looking at?
Well, it's actually been expanding. So one of the lovely things is that you never know where science is going to leave you. We proposed animal models, which we couldn't get funded. We had a group up in Canada that were wanting to use novel sort of omics type diagnostic tests on cerebrospinal fluid and to do gene expression. I was highly skeptical. It turns out, like usual, I was probably wrong at the outset. So we've been doing a lot of NMR metabolomics with a colleague out at University of California, Davis, who used to be up in Canada. And we've recently expanded that to a study of all encephalitis here at Children's Hospital, Wisconsin for the last 2 years. And we've just gotten funding to expand that to another 4 hospitals around the country. So we're doing rapid diagnostics, which also allows you to then measure what are called proxy markers or early markers of, say, effective therapeutic response. And so we're trying to get that published and funded so that we can more rapidly tell which therapies are effective and which not without waiting for the patient to ultimately live or die. And so that's a big emphasis in our lab. There is a scientist here named Marty Hessner who has been very interested in diabetes and has a very elegant gene expression assay, which is sort of an indirect reporter assay. Provides sort of beautiful filtering and amplification of the signal from a biological sample. We've been working with him and again have gotten some really gorgeous stuff coming out right now using his technique used for diabetes in blood, using cerebrospinal fluid of rabies patients from around the world. Children's Hospital has been a very big epilepsy center for a while. We do a lot of epilepsy surgeries, and we've learned that you have to take a big piece of the brain in order to have the therapy work. What that means is that you've got normal brain, which is coming out of children who are consenting to help us with our studies. And so we take the margins from those and we infect them in the laboratory with virulent wild-type rabies and then look at gene expressions. And again, we have some very interesting data. All of this is suddenly coming out in this last couple of months. And so we, again, are looking at really marked differences in inflammation between rabies virus and some other viruses, including vaccine strains of rabies. So gene expression, metabolomics, largely probably the areas we're working in. We've expanded that to other forms of encephalitis, so it's starting to spin funny places. But those are probably our main efforts. The most nice thing is that the recent Ebola epidemic got a bunch of drugs moved forward quickly to treat Ebola. And Ebola is an RNA virus, and so is rabies, and both are orphan. And several of the antivirals that may be active against Ebola are likely active against rabies too. And so we've just 2 months ago treated 2 Peruvian soldiers with rabies with one of these antivirals with a very rushed protocol that was allowed by the FDA, and with courier taking down drug to Peru for these 2 soldiers. One survived, one didn't. 50% is a lot better than 20%, but it's only 2 people. And so we're waiting for some of the analyses of the cerebrospinal fluids in those patients to come back, and then we're talking with colleagues now in Japan that have a rapid diagnostic test, bedside test for rabies, which we've been lacking. A lot of times we are flying blind with no rabies laboratory support at all in many of these developing countries where they're just not labs locally support. Leading Japanese investigator is donating about 200 of these rapid diagnostic tests, which would put into essentially a lunchbox where we're going to be sending down drug plus diagnostic tests plus some tubes to collect things in. And we anticipate sort of sending out these lunch boxes when someone has a rabies patient under care to see if we can get the proof of concept that these antivirals may work and generate a real rigorous clinical trial around the world.
You discussed how research and some of the patient care approaches for Ebola, epilepsy, diabetes have been beneficial for the work that you're doing on rabies. Conversely, is there anything that we've learned in our research, in our treatment of rabies, that has implications for other diseases where there may be some benefit?
Oh, absolutely. So one of the reasons that we're actually looking at a variety of encephalitis patients here now is that there are 3 main flavors. There's an infectious form, there's a demyelinating form where the connections essentially melt, and then there's sort of an autoimmune form, which really wasn't even recognized 10 years ago. We get different profiles by metabolomics, whereas currently we only make a diagnosis in about maybe 15%, 1 in 7 encephalitis patients. We also have a proxy marker for whether they get better or not. And so we think this is really going to blow up the field in terms of brain disorders and the like, and that we'll be able to diagnose in 20 minutes instead of 2 weeks what the patient has, and be able to show finally conclusively whether, for example, steroids work, or whether plasmapheresis works, or whether these new broad-spectrum antivirals work, because they're perfect for this kind of population too. So in that way, I think things are really spin in all sorts of funny ways. The other thing is it actually changes how you think about things. So I actually think about infections now as acquired metabolic disorders, that these are not just a virus or bacterium and a host response, all immunology, if you will, maybe some cytopathic effect, but they perturb the metabolism. And this is very similar to what's now being described in cancer too, where really immunology and cancer are all looking at metabolism now. And a rabies colleague of mine in Philadelphia, Craig Hooper, spends most of his time as a brain cancer immunologist. And they're finding that changing and treating the metabolic changes of the tumor actually dramatically improves the response to brain tumors. So I think we're more and more looking at, I look at infections now as being something where you knock out an enzyme or two and you get an acquired metabolic disorder, either too much of something or too little of something, and not just basically as a virus or bacterium with poisons. But what that's done, for example, is I now have another area of research here where I had a child with a very unusual form of meningitis who progressed despite the bug being dead within about probably 20 minutes of diagnosis. And what we figured out is this bacterium puts out a poisonous gas called hydrogen sulfide, which is famous in Wisconsin as knockdown gas. This is an environmental intoxication that happens in agriculture, in manure pits, silage, and happens in all those petroleum tankers that go by on the trains every day, where there are lots of stories of workers walking along the top of the train and getting overcome and falling off the rail car onto the ground. And if you fall into the tank, of course, you asphyxiate within about 30 seconds and are dead. So it turns out bacteria make the same gas, and rather than being inhaled from being in the wrong environment, they can produce it locally. So we now have a fairly vigorous research effort underway. We have medicinal chemists like Brian Smith in biochemistry here, where we're cloning the enzymes out of a variety of these bacteria. We actually think that abscesses are formed by a gaseous intoxication, where it's like a blast radius, like radiation or heat, in this case poison gas. It poisons your mitochondria, you asphyxiate internally, whatever little body part has the infection, and you form a hole. And so we have cloned out enzymes from a variety of these bacteria, and Brian's making enzyme inhibitors of these that are more specific for bacteria than humans. So right now, the drugs we have available are about 1,000 times better at intoxicating us than the bacteria, so we have some work to do. We have a bacterial geneticist that's helping us knock out the enzymes in some of these bacteria right now, Chris Christich. Kathleen Schmeina, who is a member of radiology and biophysics here, who has brain tumor models, is helping us with advanced 9.4 Tesla MRIs to follow rats sequentially. You don't have to sacrifice the rats, you just watch them over time like you do people. Saves a lot of rats, and we have a beautiful model of brain abscess formation with these bacteria involving MRIs in rats. And the environmental intoxication is treated with hyperbaric oxygen. We have a local expert neurologist who's also a Navy captain, knows hyperbaric medicine, and so we've been doing hyperbaric therapy on rats and are showing indications that it's actually effective. So we really expect probably, say within 5 years, to have not antibiotics, but small molecules which actually would prevent the toxicity of the infection from occurring, being able to rescue the intoxicated cells, hyperbaric, and restart the engine, if you will. And so we anticipate a lot of infections that we will be treating them entirely differently, and not just with an antibiotic within about 5 years. And so we're looking forward to that.
Yeah, you've mentioned a number of people, Dr. Schmenda, Dr. Whalen, Dr. Hessner. You've got new colleagues now.
Oh, yeah.
Who are there others? Are there people now that you're collaborating with that perhaps weren't the people that you worked with when you initially came on board?
Oh, well, I didn't know anybody when I came on board. I came on to work with Nita Salzman, who I've yet to figure out a joint project now that I've moved over, although I still admire her work. She's head of the Microbiome Institute now here, so I keep hoping we'll be able to play again. But Andreas Bayer as well in physiology. Again, the hydrogen sulfide thing, that's a potent vasodilator, recently discovered. Colleagues like Julie Fried over at Frater that are anesthesiologists have figured out that liver transplant patients get very septic without infection near time of transplant and at transplantation. And they've been using vitamin B12, which is used for carbon monoxide poisoning, which is very similar to hydrogen sulfide poisoning, and have seen miraculous responses in their patients. And so Andreas Bayer is a heart center basic scientist. He looks at vasodilation and using human vessels from fat taken out at time of elective surgeries. And so we're doing some preliminary stuff with him too, where we think we can treat septic shock with maybe something similar as a vitamin shot. So stay tuned. That's pretty early stuff. It may not pan out, but there's so many people around here. Everywhere you go, there's somebody new that's tremendously helpful. The redox biology type people too. They actually started us in the hydrogen sulfide with a little pilot grant. Jeanette Vasquez-Vivar and Neil Hogg have been strong supporters for forever. Jeanette goes back to the rabies stuff, where we have a deficiency of something called tetrahydrobiopterin, which we actually now use for treating vasospasm and bacterial meningitis here, using transcranial Doppler. So the world's a small place, and everything sort of tends to intersect. So as long as you're willing to go to a couple conferences, the world's your oyster, I think.
Now, beyond the work that you've been doing on rabies, you've also been involved at MCW in terms of the interdisciplinary program that we have, the Center for Emerging Diseases, Infectious Disease, I believe. What kind of work, and how do you work with those colleagues?
You know, that's largely been a membership thing. I really haven't worked very much with them. Dara Frank's been we almost wrote a grant together, except for our preliminary data really didn't pan out. We refuted a paper that had been published, I guess. But I think mostly where they're there is being able to talk things over. It's helpful. It's a small group. Really, there are not that many people doing this kind of research here, and so we're self-supporting. Amy Hudson works on human herpes virus 6. The gene expression stuff we're doing with rabies, we actually use her virus as a control virus. Turns out we can grow human herpes virus 6 beautifully in brain. Normally it doesn't grow in anything. So we're looking at the genes involved in that process. So you end up talking to a lot of them. Joe Barbieri and Dara Frank are close colleagues. Jen Coburn really pushes that group along. So there's a lot of mutual support. I read their grants, they read mine. But in terms of the center itself, we really haven't been able to do very much with it.
Now, we've talked about rabies as, in some respects, an orphan infectious disease, but we also see emerging infectious diseases, things like Ebola, things like that. How, as a pediatric infectious disease specialist, how do you prepare for some of these emerging diseases?
Well, you tend to read along with everybody else. I think one of the advantages of being sort of older is that you can almost always find parallels, and you can also sift through the early notices and recognize prior diseases. I've been with the HIV stuff since the beginning. You can see something coming from 100% fatal to something which is a chronic disease in the course of a couple of decades. When Ebola came out, this time, I think we were all expecting the survival to be much better than it was. We were right. It actually reads like cholera more than anything else, to my mind. So I think not being scared and knowing how to filter the reports coming out. Zika's another example. I think Zika is largely a flash in the pan. We've seen that with West Nile a few years ago. So I think after a while, you've been around the corner a few times, you can figure out where to put your efforts and where to not. The hardest part about the new diseases is being clinically prepared, and a lot of times that actually means putting your life on the line, and that's increasingly hard to do in a society now where everything is much more of a job than it is a vocation. Medicine has changed, and sleep deprivation is recognized, and you need shorter shifts, and people spend a lot of time perfecting sign-outs. I'm part of the generation where you didn't sign out. If your patient was sick, you stayed and you took care of them. These new communicable diseases, I think, are along the old model, and I think will probably to some degree bring us back closer to that fiduciary responsibility, that the fact that you have to be there for them, and you have to know them, and you have to own them. And again, with Ebola, it's interesting because you're going to have to have a certain number of people to go in. Ironically, that disease, like rabies, is largely a critical care disease. It's really not a disease where the infectious disease chops come from being at bedside. It comes from being in the lab. And so we actually made the decision that infectious diseases would not be the people in the room, that we didn't have the chops to intubate or to resuscitate fluoride shock, and that other people were better suited to that, and that our job would probably be much more behind the scenes in terms of Kelly's Hendrickson's rapid diagnostics, or our metabolomics, or Marty's gene expression, or other ways of going at the mechanism fast and furious, and with the idea that probably learning from that first one or two patients. By the third or fourth patients, you'd have a strategy up and underway, really like we did with rabies.
You know, we've talked about your work as a clinician, you know, the research that you've done. You're also a teacher, and you've had the opportunity to work with young faculty members, work with residents, with medical students. As you've mentored them, are there others that have gone into the field that have created careers that you take pride in?
You know, it's funny, here we actually have not done that much mentoring. I ran a fellowship program before coming here. Our group here has not had fellows. The field has had a glut, actually, and our sense is that probably too many were being trained, and so we actually made the decision to do more work ourselves and to not actually add to the glut. Interestingly, this year there's a shortage for the first time. So in the short term we were maybe right, but maybe in the long term we weren't. The medical students, I tend to mentor, teach as they come through. I don't have very heavy teaching loads. I have been able to broker instructors for other courses where, knowing a lot of the clinicians from practicing fairly heavily here, I can recommend neurologists or geneticists or cardiologists, whatever, for other courses too. So I think I'm probably more of a back room person in terms of a lot of the direct education, lacking really a formal training program around here. So it's fun. It's fun to talk one-on-one. I actually think that's where I do my best teaching. I think I'm probably a little more boring as a lecturer. So.
Yeah, we've talked about a number of people in a number of different disciplines that have collaborated with you. In some respects, does that mix of people from all these different backgrounds really create something unique here at MCW that's enabled you to grow that perhaps you wouldn't have found at some other institutions?
I think so. I think in kind of funny ways. I actually think the clinical care is better here than where I was before, which was a much better hospital. I think part of the reason is this is really a larger community hospital with higher volume and easier stuff. And so I think that kind of helps. I think the fact that not everybody's a world expert and not all science is really at the Nobel laureate stage already means that you have a lot of new ideas which are in the process of developing. And so I think sometimes you got to be in a smaller pond to really get the work done and to have the latitude to get it done. You know, here you can have people support you and believe in you, whereas maybe that grant's going to take another 2 years to get here. And so through a couple of pilot program support type things here, we have the Children's Research Institute within Pediatrics. We have CTSI type grants, the translational grants. We have HW grants. We have Center for Infectious Disease Research grants. A variety of really small paycheck type of things in many situations, and increasingly larger ones as people realize that it takes a little longer to get the plane off the ground. You can develop things and you can help other people develop theirs too. And really a lot of times the Nobel laureates do their work here and then they end up at the Harvard or the Hopkins or WashU or whatever else once all the good work's been done. So I kind of view that as this kind of place. There are jewels here. It takes a few years to recognize who pulls freight and who the jewels are, and then you just sort of self-aggregate, maybe like little slime moles, and you kind of make something which gets vertical after a while. So.
You've had the opportunity to work at some of the major institutions in the nation, major research institutions, as well as patient care. If you look at the culture here, is there anything about the culture at MCW that may be different than what you'd find at some of the other institutions?
Every institution has a culture, and it's really like looking at faces in the crowd. They're all interesting. They're all unique. I think the advantage here is it's probably a small enough pond where you can kind of grow things. The people here, this is Midwest. People are intrinsically polite. They don't yell at you. They don't slam doors. They don't lock bathrooms away from the other lab. And so in that sense, I think it's more collegial and maybe you get stuff done faster. be totally honest, at times it's a little provincial too. It's a little harder to convince people that it takes them longer than 24 hours to fly someplace and get reimbursed for it. So each place has its good properties and bad properties. Again, really like people. The thing that surprised me coming out of a big place is that when you look at the small places is that there are really first-rate people everywhere. And that wasn't intuitive to me, being formed in a big place. But having looked at a couple of jobs around at various times and having come here, it's evident that you can find good people everywhere.
Well, thank you. I appreciate this, and thank you for all the contributions that you've made. You've really made some significant findings that have advanced the field. And you've also helped advance the reputations of both the Medical College of Wisconsin and Children's Hospital of Wisconsin.
Well, that's for free.
Yeah.
We're in this to take care of the patients, I think. So
right.
so I'm glad that happens.
Great.
Again, it's just dessert. Yeah.
Thanks.
Thank you.